Welcome to Science News Review. For August 19, 2026, we are looking at the mechanics of biological control, from the microscopic precision of RNA interference to the systemic shifts in a shrew's anatomy, and a major medical milestone in treating narcolepsy. In the recent film Spider-Man: Brand New Day, there is a concept involving a device called a superpower inhibitor. The idea is that the device manages changing DNA by using something called short interfering RNA, or siRNA. While the idea of a gadget that can instantly dial your genetic traits up or down is the stuff of comic books, the underlying science is very much a reality in modern medicine. The FDA has already approved several siRNA treatments for various genetic diseases. These small molecules work by intercepting messenger RNA, which essentially intercepts the instructions sent by your DNA, preventing specific proteins from being produced in the first place. However, applying this to something as complex as a human's physical abilities presents massive biological hurdles. Chemical biologist Anastasia Kavarova notes that while siRNA could theoretically target mutated DNA, complex traits, like superstrength, likely involve many different genes working in concert. If you try to silence just one, you might trigger unintended consequences in others. There is also the issue of precision. Current technology struggles to tune these levels exactly. Even the idea of a switch is a bit of an oversimplification. Immunologist Judy Lieberman points out that siRNAs typically knock down gene expression rather than shutting it off entirely. This might actually be a benefit for a hero, as it could allow for a controlled trickle of power rather than an all-or-nothing effect. The real challenge, though, is delivery and duration. Physicist Shrew Wong notes that siRNAs aren't currently designed to work throughout the entire body, and a single dose can have effects that last 6 months or longer. If you wanted to turn those powers back on, you would need an inhibitor that is biodegradable, and even then, Lieberman warns, it could take several days for protein levels to return to their full strength. It's a reminder that while we are learning to edit the biological script, the script is much harder to rewrite than the movies suggest. Moving from the microscopic to the systemic, we have a significant development in clinical medicine. The FDA has approved the first drug designed to treat the underlying cause of narcolepsy, an oral medication called Orzifil. For a long time, treatments for narcolepsy have focused on managing symptoms rather than addressing the source. However, researchers have known since the late 1990s that type 1 narcolepsy is linked to a deficiency in a neuropeptide called orexin. Developed by Takeda Pharmaceuticals, Orzifil is an orexin receptor agonist, meaning it mimics that missing peptide to help stabilize the sleep-wake cycle. In phase 3 clinical trials involving 273 participants, the results were significant. Patients were able to stay awake more consistently during the day and sleep more soundly through the night. They also reported improvements in symptoms like cataplexy, sleep paralysis, and hallucinations. Emmanuel Mignot, a sleep medicine specialist at Stanford University who helped discover the link between orexin and narcolepsy, noted that the drug allows patients to experience a state of wakefulness much closer to that of a person without the disorder. For the scientific community, this is more than just a win for narcolepsy patients, it serves as a potential proof of concept for treating other neuropsychiatric and sleep disorders by targeting specific biological mechanisms rather than just masking the symptoms. Finally, we look at a different kind of adaptation, one that is driven not by medicine but by the necessity of surviving a harsh winter. In Hokkaido, Japan, researchers studying long-clawed shrews have discovered a seasonal physical change that contradicts what we usually see in small mammals. Most small animals undergo what is known as Dienel's phenomenon during the winter, which is a process where they shrink their brain and skull mass to conserve energy. The shrews in this study do indeed exhibit that shrinkage, but they have an unexpected outlier. Their snouts actually expand during the coldest months. By analyzing over 100 museum specimens collected over several decades, Yugo Ikeda and his team found that winter snouts were about 7% taller and 6% wider than those in the spring. This swelling complicates our understanding of how mammals adapt to extreme cold. Researchers have proposed 2 main hypotheses. Perhaps a larger nasal cavity houses more blood vessels to warm freezing air before it reaches the brain, or maybe the increased surface area helps improve scent detection to find food buried under the snow. While these ideas offer a fascinating look at how skeletal plasticity works, the exact biological drivers remain unconfirmed. Evolutionary ecologist Jan R. Taylor suggests that we need more research into different climates and specific nasal tissues to solve this puzzle. What we do know is that skeletal adaptation isn't always a uniform shrinking act. Sometimes, biology chooses to grow in one direction even as it retreats in another to meet the demands of a changing environment. Whether it's the precision of a targeted drug or the structural plasticity of a shrew skull, biology is rarely a simple, uniform process. It is a constant, often contradictory, negotiation with the environment.